The thing about absorption is it's primarily about transport. We need to transport the nutrients from the lumen of the small intestine into the intestinal epithelial cell, out of that epithelial cell on the other side, and finally into the capillaries to carry the blood to the rest of the body. If we take a minute to think about those capillaries, recall what type of capillaries we have in the small intestine where most absorption takes place. They're fenestrated capillaries. And what's so great about fenestrated capillaries? Those filtration pores. There are those little tunnels that go all the way through the capillary cells so that we don't have to cross more membranes. The nutrients, once they get through the epithelial cell, can diffuse easily into those fenestrated capillaries. So that means we need to concentrate on being able to get our molecules from the lumen into the epithelial cells lining the small intestine. And before we can do that, we need to break them down into small enough pieces that we can actually transport them across the membrane. So let's take a look at the main dietary molecules one at a time, starting with the carbohydrates. Carbohydrates start out as long chains of sugar molecules. Things like starch and glycogen are polysaccharides, or long chains of sugars. These are broken down into individual monosaccharides by the action of salivary amylase in the mouth and pancreatic amylase in the small intestine. And remember those brush border enzymes, the sucrase and lactase, and there are a few others that break down the carbohydrates into individual monosaccharides, like glucose, galactose, and fructose. Glucose, galactose, and fructose are very polar molecules. They're not going to cross a membrane very easily, so they need to have transport proteins. The monosaccharides are transported from the lumen of the small intestine into the epithelial cells by specific transport proteins that bind to the monosaccharide and change shape to release it inside the cell. Some of these work by just facilitated diffusion. So the sugar molecules are going to go down their concentration gradient. They'll go into the epithelial cells because it's less concentrated in the epithelial cells than in the lumen. But we also have some secondary active transport at work. If you think back to what you know about secondary active transport, that's letting one molecule go down its concentration gradient so you can bring another one up its concentration gradient. What we use to drive secondary active transport in the epithelial cells in the small intestine is sodium. Sodium is at really low concentrations inside cells. We let sodium come into the epithelial cells if we can bring along a sugar molecule with it. And that lets us absorb a lot of the sugar molecules out of the lumen and get them into the epithelial cell. Then we have other transport proteins that let them out of the epithelial cell on the other side, and then they diffuse into the fenestrated capillaries. Proteins work much the same way. We start with a long protein. It goes into the stomach and it's unfolded by the denaturing action of the hydrochloric acid, chopped up by pepsin, chopped up even further by the chymotrypsin and trypsin from the pancreas, and finally the peptidases break them into individual amino acids. Those individual amino acids are brought into the epithelial cells lining the small intestine by specific transport proteins. These specific transport proteins, the ones for the sugars and the ones for the amino acids, are pretty specific. They'll only transport one type of molecule from the lumen into the epithelial cell. Then we transport them out again on the other side, and they diffuse into the fenestrated capillaries to go out to the rest of the body. Other water-soluble things, like water-soluble vitamins, like ions and minerals, these also dissolve easily in water, and they tend to be absorbed by the same mechanism. There are specific transporters that bind to them and move them into the epithelial cell, out again on the other side so they can go into the capillaries. We see something different when we get to the lipids. Remember that lipids are not water-soluble. Lipids, like fats and oils, don't like to interact with water, so they end up being treated a bit differently. First off, we need to take the globs of fat from our diet and break them into very tiny microscopic droplets. That's emulsification. That was done by the bile. Once we've got them broken into tiny pieces, then we can break apart the triglycerides into individual fatty acids. These fatty acids, as well as other types of lipids like cholesterol and other steroids, phospholipids, all of these, since they are nonpolar, they can cross the membranes themselves without transport proteins. So the lipids, once they're broken down into tiny pieces, are absorbed by simply diffusing into the epithelial cell. They don't need a transport protein. Once we get the lipids into the epithelial cell, we have to have a way to get them out on the other side. And to do that, we package the lipids up into little packages called chylomicrons. These chylomicrons are put together into a vesicle and released on the other side of the epithelial cell by exocytosis. The chylomicrons are now floating around on the outside of the epithelial cell. They are too big to fit into the capillaries. Instead, they get swept up by the lymphatic system. Remember, we talked about one of the functions of the lymphatic system being to absorb lipids from the digestive tract. These chylomicrons, once they're into the lymphatic vessel, get carried through the lymphatic system, and then they're dumped into the subclavian veins, and that's how we get lipids into the bloodstream. Other fat-soluble things, like the fat-soluble vitamins, I mentioned cholesterol, all of these sorts of nonpolar molecules are absorbed the same way the lipids are. They're all going to diffuse into the epithelial cell, be packaged together into these chylomicrons, released by exocytosis on the other side of the cell, and swept up by the lymphatic capillaries. The last thing that's really important to talk about absorbing is water. Altogether, between the water that we drink and the water that's in our food and the water that's part of all the secretions that we're pumping into our digestive tract, about 9 liters of water goes into the digestive tract each day. 9 liters does not come out the other end. Very little water comes out through elimination. Most of the water is absorbed in the small intestine. About 8 of those 9 liters will be absorbed in the small intestine. And it's absorbed by the process of osmosis. So remember that in osmosis, water is attracted across a membrane to an area of higher solute concentration. Well, as we are absorbing all the nutrients out of the lumen of the small intestine, the water is attracted to those absorbed solutes and is also going to be moving by osmosis into the epithelial cells. Now there's something interesting about what happens to the absorbed nutrients in the blood vessels in the small intestine, because they don't go right back to the vena cava to go out to the rest of the body. The capillaries in the small intestine, in the digestive tract, they collect the nutrients and then they drain into a vein called the portal vein. The portal vein does not go back to the vena cava. Instead, it goes to the liver. So almost all of the nutrients that we absorb from the digestive system go to the liver before they go anywhere else. Think about why that would be important. We consume a lot of things that aren't all that great for us. A lot of toxins come in with our food. And the liver is good at taking care of toxins. So we take all this food that we just absorbed and all the other things we might not want, we send into the liver. The liver can take out the toxins and other molecules we don't want before sending that blood on to the rest of the body. The liver also serves as sort of a special storage spot. The liver helps to store glucose, and it stores a lot of amino acids and also vitamins and minerals, and whatever's left then goes on to the rest of the body. The blood from the liver is collected into the hepatic vein. The hepatic vein drains then into the inferior vena cava and takes that nutrient-rich blood out to the rest of the body. While we're talking about the liver, I want to make sure that we don't lose sight of the fact that the liver is a really important organ that is involved in a number of different body systems. The liver is important because in the digestive system it removes toxins and pathogens from the blood, including those that were absorbed with the food. It stores glucose and fat-soluble vitamins and other nutrients. It also has the enzymes needed to break down fats and amino acids for energy. Not all the cells in your body can do that, but your liver can break down fats and amino acids for energy that can then be used by other cells. Your liver also has the ability to synthesize glucose. We talked about that earlier. And the liver can also make fatty acids, cholesterol, and some of your amino acids. So a lot of the nutrients your body needs can actually be synthesized right in your liver. We also talked about the liver producing the bile that we need for emulsifying fats for digestion and absorption. When we were talking about the circulatory system, the liver was very important because it synthesized so many of the plasma proteins in the blood. Things that were important for maintaining blood pressure, important for blood clotting, proteins like albumin that we need for transport, alpha and beta globulins that are important for transport and clotting and affect what's going on with viscosity and osmolarity of the blood, the fibrinogen that's important for clotting, angiotensinogen that we need for regulating blood pressure. All of those are important proteins made by the liver. And we also talked about the liver being the location of breaking down red blood cells. So that's a really important organ that does a whole lot of important things. Finally, once all that food has worked its way through the small intestine and all that's left is just the food residue, it gets to the large intestine. The large intestine contains our normal flora, and these have the ability to break down some of the other molecules we couldn't break down ourselves, so we can do a little bit more absorption, get some more nutrients out of those. The normal flora also produce some of our B vitamins and our vitamin K, which makes vitamin K deficiencies rare unless there's something wrong with your large intestine. The large intestine also contributes a bit to absorption. We have to absorb the vitamin K and B vitamins that our normal flora made, or there was no point in getting them. We also reabsorb a lot of sodium and chlorine from the large intestine, and we absorb more water as well that's attracted to those sodium ions that are being reabsorbed.